All right, so let's talk more about the microscopic structure of the brain. And to be specific, we're going to go over the general features of neurons and how we attempt to classify them, or have attempted in the past to classify them, and also the far more numerous cells of the central nervous system, the glial cells. We'll go over those as well. Now, at the end of the last lecture, we concluded talking about the reason why we consider Santiago Ramon y Cajal to be the, quote, father of neuroscience, end quote, and not Camillo Golgi. And remember, it was Cajal that promoted the idea that the brain was no exception to cellular theory, that it was composed of individual units that were later dubbed neurons, and that communicated by contact with one another. And again, that was finally proven in the 1950s. But that idea that the brain was composed of individual units that we call neurons came to be known as the neuron doctrine, and he was the promoter of that. And this is one of his drawings. I showed this last time, but I didn't mention what it was. This is a slide of cortex, I believe from a rat, that has been Golgi stained, and this is Cajal's camera lucida drawing of that. So he would have had a fixed slide of the cortex that had been Golgi stained, and he would focus up and down and use the camera lucida to trace out, or to draw out, all the extensions he saw. And this is one of those drawings. You can see the stamp if you look carefully. It says Museo Cajal, Madrid. So this is a picture from a museum. Now, there are a few of his camera lucida drawings in private hands, but most of them are still owned by the Cajal family, but they are considered as works of art as much as scientific artifacts. All right, so let's look at a typical neuron. Now, even before the silver stain and that allowed us to see all the different structural complexity of a neuron, people noted that there was a central body, which in Greek is called a soma, which just means body. Another name you'll see for it is perikaryon. That's not used very much. That literally means around the seed or around the nucleus. So if you'll note here, this is a Nissl stain, and that's really all you can see in a Nissl stain is the region around the nucleus, and so it became known as the perikaryon. Okay, so you have the soma, and then coming off of the soma are various neurites. That was the general term given for them. Later, more specifically, they were named dendrites and axon. Always, or often, not always, but often multiple dendrites will be emerging from the same soma, but always a single axon. So there's always only one axon emerging from the soma. Now the axon may branch later, what we call axon collaterals, but as it emerges from the soma, it is always singly. Okay. Now it wasn't really clear, all this wasn't that clear from the Nissl stains until we got better stains. So that earlier name neurite simply referred to the many extensions that could be seen emerging from the soma. Okay, so what I'd like to do is go through the structure of a typical neuron and look at its different features, and then we'll go in and look at different ways they're classified as well. Okay, so this is a drawing I made some time ago where I divide a typical neuron, or drawing of a typical neuron, into 3 zones. An input zone, and I call it this because this is where a neuron collects information from other cells. there are a number of ways to model the function of the entire nervous system or conceive of it. One way that's attractive, it's not entirely accurate, we'll talk more about that later, but an attractive way to model the function of the nervous system is to view it as an input-output system. So data comes into it, gets processed or integrated by the central nervous system, and it goes out, and then some output occurs. So the input you might view as sensory information, and then the output you might view as motor information, like movement. It's not always like that, but movement, okay? Now, one of the reasons that's such an attractive way of looking at the nervous system is because it's actually what every neuron does individually. So in that way, then, the entire nervous system is simply a reflection of the activity of individual neurons, magnified by this enormous complexity, because there are perhaps 100 billion neurons in the brain and spinal cord, and they are connected to each other, not in random ways. There are definite patterns of their connection, but if you were to add up all their connections, you're talking, it's countless trillions of connections. So this enormous complexity simplified to input, integration, output, and then reflected in the same basic function that we see in each individual neuron. Now again, something that attractive is not probably going to end up being right, and so it isn't quite. But nevertheless, the activity of a neuron is somewhat this simple. Now, of course, it's biology. There's very few absolute rules in biology, and there are exceptions to what I'm going to tell you here. But it is a nice, simple, basic way of looking at the function of a neuron. Okay, so input zone would be the dendrites and the soma. Now, the reason we call it that is because this is where inputs are coming in from other neurons. These will occur at points of contact. As Cahal said, we now know those to be synaptic connections. And we know that the method of communication there is chemical. In other words, for the most part, now again, there are exceptions, there are electrical synapses in some instances, but for the most part, what's happening at those points of contact is one cell is releasing a chemical message that is detected by the receiving neuron that's collecting the information. Okay? So the input zone. Now, I could also rename this the input and integration zone. Of course, that didn't. There you go. So what's happening also, in addition to inputs, is we're integrating those inputs together, and we'll talk about how that occurs as well. And the result of that integration will be that we may or may not send a signal down the axon, right? So that either occurs or it doesn't. And we'll talk about why that's the case later on. So there's a signal, it's called an action potential, you may be ahead of me here, that either does occur, or doesn't occur in response to these inputs. And once it does, it goes down. If it does, it goes down this axon, the conducting zone. And that is a fairly straightforward process, and it's very reliable. Your brain, or the function of your nervous system, assumes that conduction down axons is going to be reliable. Now, once the signal is conducted down the axon, we reach the output zone, which is where this neuron will in turn form points of contact, synapses, with mostly other neurons, though sometimes with output structures like muscles, and where it will send its information on. So the output zone of one neuron feeds into the input zone of another. Now, one other thing I want to mention is that the reason that I include the soma in the input zone as well is because not only do synapses form on the dendrites, but also on the soma. Most of them on the dendrites. And we'll see why in a couple of slides. So this defines a direction of information flow. The chemical information, the chemical signals impinge onto the dendrites and soma. They are collected there. Now, before they can be integrated, they have to be converted into electrical signals generally. So they're converted to electrical signals, integrated together, and then that information flows out along the axon as an electrical signal and into the output zone, where again it's actually converted back into a chemical signal, because the electrical signal that has traveled down the axon causes chemicals to be released from these axon terminals. Right? So that's the overall view of this. Now let's look at each individual part and say more about each individual part. We'll begin by talking about the soma. And of course we mentioned that the soma was the first place part to be noted, because it can be seen in the Nissl stain. And the reason it can be seen in the Nissl stain is because the Nissl stain, again, stains for nucleic acids. This is the site of the nucleus. It's the site of the rough ER and the ribosome, free ribosomes that are going to have RNA in them and therefore would pick up the Nissl stain. And so you see that if you look at any one of these Nissl stain neurons over here, that stain proceeds a little bit up into some of the dendrites. It actually doesn't go very far at all into the axon. It's maybe just the very beginning, if at all. That's one of the features of an axon. It lacks any of that Nissl. It lacks any what were called Nissl bodies, or anything that would stain with a Nissl stain. Okay, so often we say that the nucleus or the soma, in addition to being an input, here you can see a synapse forming on the soma in this drawing. In addition to being a point of input, is also the sort of the life support system. It contains the basic functions of life for the cell. Okay. Now, often the soma is very small compared to the dendrites. If we look back at this initial drawing I showed you, they often draw the somas as very large. And in this one also, the soma is the obvious center. And it's not surprising, because that's what we initially saw, right? The Nissl stains made it look like it. And in some neurons it is like that. But in many neurons, most of the surface area of the cell is in the dendrites. That's where most of the inputs are. And the soma is almost lost in that. Okay, so here we can see a couple of fairly accurately reconstructed cells, neurons, a stellate cell and a pyramidal cell, that we can then see just how elaborate the set of dendrites are, and how almost lost the soma is in that mass of dendrites. Okay. Now we come to the conducting zone. I'll come back to the dendrites later. Now, again, you always have a single axon. It may or may not be myelinated, and I'll talk more about myelination later in this lecture, and we'll talk about why an axon would be myelinated when we get to talking about action potentials, right? But there's always a single axon, and the action potentials, or the electrical signals, are going to be generated at this region here where it begins to slope away, okay? And that's called the axon hillock. So let's see if I can. So the axon hillock is this portion right here, okay? Where I'll just kind of move it right over there, where the axon is starting to taper, or the soma is starting to taper into the axon. That'll be the final point where the information is integrated together and where we generate that electrical signal. Okay? Now, the unique thing about axons, in addition to not having any nucleic acids and so forth, or no ribosomes or ER in them. Another feature of it is that the membrane in it is going to have a unique set of proteins. Now, we're going to say a lot more about the membranes of a neuron. In fact, there's 2 more chapters after this one just on membrane properties of neurons, because that's a very important part of it. But one of the unique things about the membrane of a neuron is that it has a unique set of channels, and we're going to talk about those channels, ion channels, that allow it to generate an action potential. Okay? And again, we'll talk about all that mechanism, so don't get upset if you don't understand what that means now. That set of proteins allows a signal to be not only generated but regenerated. So an electrical signal is generated and then regenerated continuously so that signal travels down the axon from one end to the other. And again, it does it reliably, and it does it over any distance that's needed. And it often does it fast. The fastest axons in your body, the conduction velocity in them approaches around 300 miles an hour. And if you try to figure out how long does it take to go between any 2 parts of your body, if you're going 300 miles an hour, the answer is not long at all, right? So works pretty well. Okay, to help show you that, I have a picture of a blue whale here. Now, blue whales are an outline of a blue whale. You may know, of course, that whales evolved from 4-legged land-based predators that presumably had some sort of a coastal lifestyle and then eventually become totally aquatic. And so whales are mammals, and they have the same basic outline, or their nervous system has the same basic layout as a human's. So the longest axons in our bodies are sensory axons that carry senses from whatever the longest toe is. For most people, it's the big toe. It could be the second toe, right? So if someone tickles you on the end of that toe, that's going to generate action potentials that will travel all the way up to your brainstem. And so that's going to travel nearly the whole length of your body. So a blue whale is going to have a similar layout. So if you were to tickle the blue whale on the end of its fluke there, action potentials would travel all the way up to its brainstem. Just above its eye right there. Okay, so that's a long way. And it does that reliably, right? So, and quickly. And so we're going to talk about the mechanism by which that happens. Now, one thing I want to mention is that the mechanism of a neuron releasing a chemical at the end of its axon and then going on to a cell here, another cell, is not unique. That is how cells communicate. When I teach this in anatomy and physiology, I say, how else are cells going to communicate? It's not like they have, and nearly everybody in the class immediately gets the stupid pun I'm about to make, and they say, cell phones. So, okay, you can groan if you want to, but again, this is how cells communicate. If they're not in direct contact with each other, that one of them has to release a chemical that the other one detects with a receptor. And so that's not unique. The difference with a neuron is that most of the distance between the 2 cells, if they're far apart in the body, is covered by this structure. It's an extension of one of the cells, the axon. And so the distance the chemicals have to travel is very, very, very small. And so that means it's very, very, very fast. Okay? Contrast that with something like an endocrine hormone, which has to travel through the whole bloodstream, and you're talking about a millisecond scale communication in the case of an axon versus tens of minutes or minutes in the case of an endocrine hormone. Okay? Now, one of the consequences of having that very long axon is that all that material that's in the soma that needs to support the axon has to go a long way. And in order for any material that's produced in the nucleus, right? Remember, we've got to, that's where proteins are going to be made. So anything you got to make has got to be made there, protein-wise anyway. It's got to travel all the way down the axon if it wants to get to the axon terminals. So how do we do that? Well, we couldn't rely on diffusion for that, random diffusion. You could maybe in a small cell that wasn't all elaborate like this, like a bacterium, but you can't rely on that in a neuron, certainly. And theoretically, it would take years for something to diffuse down some of these axons. And so, of course, we have a directed motion that relies on the cytoskeleton of the neuron. Now, I'm not going to go into a lot of the basic cell biology stuff that Chapter 2 covers about neurons, right? Neurons are cells. They have all the parts of a cell that you will have learned about in other classes. But I do want to mention that within the axon and within the cell generally, but especially within the axon, there is a network of microtubules that are going to be used for axonal transport. Okay, we'll talk about those briefly. You also have a class of intermediate filaments called neurofilaments. And if you know something about intermediate filaments, they tend to be much more permanent and rigid than either the microfilaments or the microtubules. And so those are a big part of what helps the cell maintain its unique shape, are those neurofilaments. But the microtubule network is going to be responsible for axonal transport. Okay, and so here we can see some vesicles that presumably contain proteins that we're going to need. And they're attached to kinesin, a motor protein, and they're going to be crawling from the soma where they were produced and moving down the axon. And they can go somewhere like 1 to 10 millimeters a day, so almost pretty much a centimeter a day. So now you can, it still takes a long time, but now you can move down that axon in a reasonable amount of time for the life of the animal. Now, that long axon I mentioned that goes from your toe to your brainstem, the soma of that is way in the base of your spine, the bottom of your vertebral column. So, it doesn't have to go quite as far. And that would be true in the blue whale as well. Boy, that's hard to say. You may, if you've taken cell biology, you may know that there's another molecule that microtubule transport is directional. So kinesin can only go in a certain direction. And the way that this is arranged, kinesin molecules are going to carry or transport material in what we call an anterograde, meaning from the soma to the axon terminals. That's the normal direction that information flows, so we call that anterograde. There's another transport molecule called dynein that can move in the opposite direction. So sometimes things need to go from the axon terminals back, and dynein will move them along the microtubule network in that direction. Okay, now we can actually take advantage of, that would be called retrograde transport, right? So dynein would be responsible for retrograde transport. And we can take advantage of that. This is a well-established technique. Is that if you have a target site, you know there are axons going into this location, but you don't know where the neurons that produce them came from. So you're trying to trace a circuit, right? You know where the circuit ends, but you don't know where it began. You can inject some sort of label in the area. Now this has to be done in a living animal. In this case, it's a dye called horseradish, or an enzyme called horseradish peroxidase, that after the tissue is fixed, you can expose it to a substrate, and it will produce a colored substance that you can see on the slide, right? So you inject horseradish peroxidase, and it gets taken up by the axons. Usually as they're recycling membrane, they'll take up some of this stuff, and it'll get retrograde transported back to the soma. And so you can, after give a few days for that to happen, and then you can sacrifice the animal and put make slides of that tissue, and then you can see where the HRP stained cells are, and you can see where this is done. You can do this also with living animals, with fluorescent dyes. When I was just beginning grad school, I did some experiments where we were trying to identify neurons that controlled the tongue in rats. And so we injected a fluorescent dye under the tongues of these rats. And then that dye was taken up by the nerves that controlled those muscles. And then when we sliced up the brainstem, we could see where the fluorescent dye was. Now, the slices we were making were still living. It's possible to take out the brain of an animal, keep it alive long enough to slice it up, and then have living neurons in those slices. And there's that can help you study their function. And that's what we were doing. So we were able to see this glowing red cells, and that's the ones we targeted with our electrodes to study their function. This is also used, anterograde and retrograde transport are also used in some pathological circumstances. So for example, when an animal is bitten by a rabid animal, the rabies virus enters nerve terminals and of sensory neurons and travels in an anterograde fashion up into the central nervous system of the animal, where it infects the brain, causing all the behavioral changes we're familiar with the rabies, and then travels back out axons to enter salivary glands, so which can then infect another animal. This also happens with herpes. Herpes, when it infects your skin, travels up axons and then infects sensory neurons, and then later on, if you have an outbreak, the herpes viruses will travel back out in an anterograde fashion, travel back out the axons, and then erupt from the nerve terminals, and that causes the characteristic inflammation of the herpetic lesion. Here we can see someone with suffering a shingles outbreak, and those would be herpes zoster lesions on his neck there. This also happens with some bacterial toxins. Tetanus toxin travels from the site of a wound where the Clostridium tetani bacteria are infected, and the toxin will travel up a retrograde transport to the spinal cord, where it then crosses actually synapses and damages neurons that would normally inhibit movement. And so that's why you have this uncontrolled spasmodic movements in an acute tetanus infection. This is a famous patient of a human patient dying of a tetanus infection. Okay, so now we come to the axon terminals, or are we in the dendrites? Yeah, so let's talk about the dendrites now. The dendritic tree. Dendrite, of course, is from the Latin for branches. That's where we get that. And the collection of dendrites is sometimes called the dendritic trees, right? And they can vary in complexity. And so we can see something relatively simple like this motor neuron in the spinal cord to something very, very complex like Purkinje cell. Showed you one of those before. So these can vary enormously in complexity. And again, sometimes the soma is quite small compared to them. Some of the dendrites will have, I don't know why I have this extra one, I guess just to show you the typical arrangement of the stellate cell there versus the pyramidal cell. We'll talk more about these shape classifications in a moment. This is one called a chandelier cell, I believe. Sort of looks like a chandelier hanging down. Some of the dendrites have spines on them, small sub-processes. This is a reconstruction of the apical dendrite. So this is the pyramidal cell, very typical cell you'd find in the cerebral cortex. And you can see this single dendrite coming up called the apical dendrite. And it has spines on it, sub-processes. Those spines are thought to be chemical compartmentalization, have the function of chemical compartmentalization. Because as we'll see, the communication between cells is not just a matter of electrical signals, right? We know it's converted to a chemical. But the way that chemical is received varies. In some cases, it merely causes the opening or closing of ion channels, which results in a relatively simple electrical signal. In other cases, it results in a more complex biochemical response. And so it's thought that the spines allow for compartmentalization of those biochemical responses. So you could have, for example, a synapse that occurs directly on the shaft of a dendrite, and then one that occurs nearby in the spine, and chemically those two, even though they're close together physically, chemically they can be quite different. That's thought to be the function of spines. We do see disruption of spines in certain conditions. So here we can see normal spines in this case, and then disrupted spines in someone with some various form of mental impairment, right? So various forms of impaired mental ability, usually from birth, in various forms. We see that many times reflected in, or one of the anatomical changes you can see if you were to analyze the brains of that person is a disruption in the density and the lengths of the spines. They don't look quite the same as someone who isn't afflicted by mental impairment. Also, we think that this is a consequence of lifelong use of some antipsychotic drugs. So here you can see normal brain of an elderly person, and here you can see the brains of an elderly people that were schizophrenics and spent their lives on taking antipsychotic drugs. And so you can see quite a profound disruption of the spine. Density. We're not sure if that is a result of the disease process of schizophrenia or a result of the effects of the drugs. But nevertheless, you can see that disruptions in spines are associated with certain mental conditions, either mental intelligence impairment or a lifelong neuropsychiatric disorder. Okay, so then we come to the axon terminals, and we'll have a lot more to say about these when we get to the whole chapter on synaptic transmission. But basically what we have here is the end of an axon. Now here we're showing the end of a single axon. Very often these will be branches, and you'll see axon terminal, also called an end bulb or a synaptic bulb. Sometimes you'll see that form. Sometimes you'll see it form and then the axon continue, and you'll see another one. It's called a synapse en passant. Anyway, we'll get to all that. But the basic parts are here that you have this end of the axon, this axon terminal or presynaptic terminal or end bulb. There's all 100 different names for it. And inside of that, you'll have vesicles of neurotransmitter, and these will be released when signaled to do so. These will be released into the very narrow space between this cell and the next cell, called the synaptic cleft. And then that postsynaptic cell will receive this input, in most cases, on its dendrite. Okay, though as I said, somas can also be part of the input as well. But in most cases, it's going to be received on the dendrite. Okay? But again, we'll have a lot more to say about the structure of those. I just wanted to get the basics of this down for now. We'll see a lot more about the structure and the function of those axon terminals as we get into the chapters on synaptic transmission. Okay? So I'm going to stop the video here and put the rest in another video.